Hello, everyone, and welcome to the 40th Annual Spnhc Conference. This is session CTRB03.2 entitled Digitization, Data Management, or Data Integration, and I'm your moderator, Joanna Corey-Mancha. This year's meeting was made possible in part by the generosity of our sponsors, as shown here. And as a reminder, this session will be recorded for later viewing and posted to the SPNHC YouTube channels. Thank you for joining us, and thank you to our speakers. Each speaker will have 15 minutes to present. We have some in person and some virtual. And whenever you have 3 minutes left, I will stand up. That would be a good time to wrap up for questions if you can. But of course, we're all human. We'll also be taking Zoom questions. And anyone on the Zoom, please use the online chat judiciously. And of course, if you'd like to continue the conversation online, continue using the chat function in Oxford Abstracts. With that, everyone, please enjoy the session. We have 7 speakers today. And up first is Molly Schools.
Hi, everybody. My name's Molly Schools. I'm a postdoctoral researcher at Omaha's Henry Dorley Zoo and Aquarium in their Conservation Genetics Department. And today I'm here to talk to you about an R package I designed called Taxo Tools that's designed to help facilitate taxonomic revisions and descriptions. So this work was really born out of my PhD work. During that time, I studied a group of Caribbean lizards, a family. When I started my research, that group had 2 genera and about 50 species. When I finished my work, that group had 11 genera and about 94 species. So it was a huge body of work. It was a lot of fun for the most part. I really enjoyed it. On the back end, though, that meant that my dissertation was 995 pages long. So as I was writing that, I was really seeing it was quite frustrating because the data in that document, which was basically 2 long monographs in a paper, were so nested, where I was referring to the same species, sometimes upwards of 1,000 times, sometimes the same range of morphological traits I was mentioning over 100 times. And that got even more frustrating when, of course, a committee member suggests I redo how I classify juveniles. So suddenly I'm removing specimens, adding specimens, changing which traits I can use. Or when someone discovers a new genus in my family when I'm 3/4 of the way through writing this. I've never hated scientific progress more than when I was having to Control F through this document at 11 p.m. the night before I told my committee I'd get it to them. If that ever happens to you, don't worry, because to quote my advisor, nobody's going to read the damn thing anyways. But once I had more brain power and a little bit more time on my hands, so that was when I got to my postdoc, I sat down and developed Taxo Tools, where I basically automated every single part of my dissertation that I could probably think to automate. So here we have kind of the workflow of what a traditional description might look like, where you start with your data. Most often my data was phylogenies, traits, these were mostly morphological traits, and metadata, including latitude and longitudinal points. Taxo Tools then has functions to help facilitate cleaning and processing all of those different data types before going down into some phylogenetic, trait, and geographic analyses. Of course, it's going to be up to the researcher to make taxonomic decisions. I'm not going to pretend that that's something I will ever be able to automate. But once a researcher has made those calls, Taxo Tools can help facilitate the documentation. I really didn't want to just throw functions at you guys, but this program is all functions. So I'm going to go through kind of the 5 sets of functions and a few examples of each of them in the form of a case study. Something kind of interesting about Taxo Tools is because it isn't really a traditional workflow. If you hear just 1 or 2 functions that you think would be useful to you in your research, you can go in and just use 1 or 2 functions and it shouldn't be a huge hump to get over. But so there are somewhat 5 classes of functions within this package. Each function has a prefix and an underscore before the function name. I did that just so they all appear together in the documentation. Or if you're in there just trying to use one function, it's easy to see what other ones you might already have data ready to be plugged into. For this case study, we're going to be looking at the genus Ceresia, which is a lizard group. So this was a genus that had 1 species when I started my work and 8 when I was done. There are going to be 3 forms of data we're using: phylogenies, and there are 3 of them: a maximum likelihood, a Bayesian, and a time-calibrated phylogeny. There is 1 CSV that has metadata, so collectors, dates collected, localities. That same CSV also has morphological measurements, and it has latitude and longitudinal points in it. And then 1 final CSV with old, not taxonomically updated names, and 1 column with the nice, pretty names that I would want to update these to. Starting first with kind of the data processing functions. So there's a function where you can check missing data and see what percentage of data in each column is missing. This is really important if you're seeing morphological traits with missing maybe 75% of their data to really look at and evaluate if you want to use that. You can match names between data sets, so between phylogenies and CSVs, phylogenies to phylogenies, and CSVs to CSVs. You can remove juveniles by basically putting in a user-defined cutoff and a trait. It will count the individuals within a given percentage of the largest individual in that trait as adults, remove the others from the data set, and tell you how many it removes from each grouping. And you can standardize traits by a reference trait. In the phylogenetic analyses, you can generate histograms using time-calibrated phylogenies, and you can do that with multiple groupings within that same tree. So here we have 3 species from 1 tree in 1 histogram. You can highlight taxa and nodes, and this can be by names or parts of names or support values. This one, I love this one. So you can plot node information from multiple trees on 1 tree. This is one of the few things that is a series of functions in taxo tools, but what we're looking at here is my maximum likelihood tree compared to my Bayesian tree with matching node supports painted on each node. So if a node matches 1-to-1 for the species contained between the 2 trees, it can paint the support values from both trees on that node. And finally, you can do node support histograms, and you can do that with 1 or multiple trees to compare them. For trait analyses, again, for most of my work, I use morphology, so that's what I'm looking at here. With 1 function, we can compare traits, looking at both scatter plots and interactive box plots. We can identify groups that aren't separated by a given set of morphological traits. And we can easily summarize traits. For geographic analyses, we can plot distributions and map convex hulls on top of them in an interactive map. And in a table, we can print which groupings have overlapping distributions. So those are all false because none of them are overlapping, but it would be true if any of them were. Finally, for documentation, so you can identify standout traits, which is really important if you're trying to find diagnostic traits. Using a user-defined cutoff, you can say if a given grouping differs from more than 50 or 75% of the other groups within the data set. You can generate appendix sections, which can just be copied and pasted into Zootaxa manuscripts. You can do description sections for each species where it prints out those morphological ranges for each grouping. And you can do diagnoses sections where it pairwise compares every single group and only prints those morphological ranges that are not overlapping for each grouping. So this is really important. This would have saved me hours of time and headaches in my PhD. And at the same time, when you have something automated like this, there's just less likely to be mistakes in these giant documents that are showing up. In future directions, I would like to preface that I'm a biologist first and a coder second. So as people start poking around in this, I'm sure everyone will find ways that functions could be run a bit more efficiently or have a higher degree of flexibility built into them. Also, going off of the idea of flexibility, this is coded for how I do my revisions. So I'm really excited to kind of see people that have data formatted a little bit differently, how we can better accommodate that. and different kinds of data. Specifically, I'm really interested in integrating environmental and ecological data into this. That is all. I would love to preface too, I spent more time on the documentation for each function, the vignette that goes along with the R package, and annotating everything I possibly could. So hopefully that for someone that is not a coder, this would be easy to use. And I really hope you find it that way. I have the GitHub with a beautiful vignette linked here and my resume. 2 weeks ago when I wrote this, my postdoc contract was dangerously close to ending, but I did just get a new job last week. But my contact information is there if anybody wants it. So thank you. Any questions? Yeah. Or yes, it uses coordinates formatted as decimal degrees. So there's also a data cleaning function where if you have degrees, minutes, seconds in any of your columns, it'll point those out too.
Molly, thanks. That you gave me flashbacks to my revisionary work in graduate school. Amazing stuff. I'm wondering if you did anything to facilitate the production of a Darwin core file that you could then dump as a data paper or as supplementary material for your revision that could make it right into GBIF, for example. So do you have a function that is Darwin core export star?
I do not right now. That was one that was kind of on my radar, as I would love to add it. I would also hope down the line for this to be kind of collaborative, and I would love to work with people on kind of these sorts of ideas and making it just more of a resource for people in taxonomy. And that would be a great one to start with.
Job give you time to keep working on this?
Yes, ideally.
Awesome.
There's a question from online. Will this work with non-vertebrates? I'm curious if this could work on fungi.
Ideally, I mean, all I've worked on are vertebrate species, so I'm sure I have some biases that I'm not accounting for, but anything that you're using phylogenies, you're using traits, you're using locality data for, this should be easy to adapt.
Hello there. A quick question for you. Actually, two-part question, if you don't mind. The first question is, do you actually import the phylogenies directly into the open source platform?
Yes, I do. I also, in the vignettes and in every sort of annotation for each data file, talk for a long time about what exactly I did to every single phylogeny, what program I ran it through, and what I did, like if I imported it into Figtree to first root it before taking it into another function. So that is spelled out A to B, kind of.
And the second quick question, there's a lot of studies, obviously, that have been published over the last, especially over the last 10 years or so, where people have identified species based on primarily morphological features. I'm wondering if you've considered, maybe you have, considered using your open source platform as a way to verify, what's been published using morphological data only versus what you would actually find using your packages?
My package really doesn't make those calls. So I think that that's kind of that part that is missing that I really want left up to the researcher. Mine's just kind of other ways of visualizing and exploring the data. So what I could see happening is like in an instance like that, which maybe the bulk of people would not agree with a species definition, you can kind of look in here and look at the histograms and be like, whoa, all of those divergent states are really recent. Let's re-question that. But it wouldn't actually be like, this is wrong, this is right.
That's about all the time we have for questions.
Thanks so much.
but-
Thank you.
Okay. Next up, we have Richard Dodd, who's going to be telling us about integrating botanical and mycological collection data.
Hi. Firstly, just like to say thanks for actually being here. Friday of the conference, night after the banquet, and you're here at 8:30, eager for more. So thank you. Anyway, I'm Richard. I'm from Kew Gardens. We're part of the Integrated Collection Management System team, and I'm going to talk to you a little bit about some of the changes we've made to manage taxonomy within our system. It's worth highlighting I'm not a taxonomist, I'm not a curator, I'm an IT guy. So I'm here particularly looking at it from a systems perspective and how we actually made it work. In the next 10 minutes, hopefully I can take you through some of the measures we've taken to help us with taxonomy in the system. So very briefly, I think we've probably seen a similar slide in other presentations. Kew's been building collections for the last 260 odd years. We have collections ranging from herbarium, fungarium, but through to other collections such as the Millennium Seed Bank and economic botany. All of these collections we've tried to pull into our integrated system, and each of them has brought with it its own challenges that we've tried to incorporate in the one system. We've had two major projects have been running at Kew in the last 4 or 5 years. One is the Digitization project, which is just digitizing Kew's roughly 8 million fungal and plant specimens, and then the CICMS project, which I'm a part. We're using the Earthcape platform for our integrated system. Integration is the key word there in the name. It's all about bringing together our legacy systems into one platform, partly so we can share data between them more openly, but also from a supportability point of view. Most of these systems, the legacy systems, were reaching end of life and needed to be moved for supportability and security. So each of the collections that have been moved into our new system has brought with it its own challenges, and they've each needed something a bit different. So just very briefly, if we look here, we have a herbarium object. We're obviously very keen to know about taxonomy, but also collection information and dates of that. But we can look at the same species in the seed collection, and for this object, we now need extra information. We need to be able to record information about processes that we carry out on the object whilst we have it, things like cleaning, counting, viability and germination tests. And then looking further, same species, this time in an object belonging to the economic botany collection. And here we're interested in provenance, donor information, again, history and taxonomy. Critically for economic botany, one of the key differences here is that an object in the collection can be made of more than one species. So we've had to accommodate that in the way we handle occurrences. So one of the most recent collections we've moved into the system is the living collection. And it's this collection that has given us a significant increase in what we need to manage regarding taxonomy. Hardly needs saying the taxonomy is essential. We need to be clear about our taxonomy for research. We need to know what we're talking about. We need to be able to apply regulations and restrictions correctly based on species. And for the seed bank collection, we need to be able to apply correct germination storage procedures for those particular species. Bringing together things into one system has had its challenges regarding taxonomy. The legacy systems often didn't use controlled lists, and so we have lots of cases of inaccuracies, misspellings, duplicates in those systems as we've tried to migrate them into one system. The sheer complexity of taxonomy is the next thing. This taxonomy is not easy, and then we've brought the living collection in, which has added to that burden for us. So we'll talk a little bit about that. And then when you think you've got things in place, things change. Re-identifications happen either on the individual specimen level or on gross mass recreations. So they're the challenges. What have we been doing to try and address those? We start from a solid base as much as we can. We used the world checklist of vascular plants and the Index Fungorum as our backbones. As we've moved legacy collections into the new system, we will match against those. Matching is not always going to work exactly 100%. So we have also a mechanism to manage matching to the most current taxonomic name, but also keeping that history of names used on the original specimen. So it's a little bit small here, but if hopefully you can just about see, we have an object here from the herbarium. It has a taxonomic name, but we also have fields for some of that history about the name. We have a verbatim taxonomic name towards the bottom with a reason for that change, a non-Linnaean field, and a vernacular name. All of this provides us some history about the name, even though we're still using the current taxonomic name for reference in the collection. The point at the bottom of the slide talking about identification, as I was saying, things change. We manage this by having the concept of a current identification whilst also maintaining history of previous identifications. So here we have a living collection object. It has a taxonomic name. If you notice at the top at the middle, we have a tab of identifications with the number 4 against it. And if we look at that more closely, we have 4 identifications for this object. The top one is the current one, so that's its latest identification. But we also have information about its previous identifications and the history around them. And each of those we can see why that identification was made and significantly why it changed. So we've given ourselves a solid base to start from with the backbones. We have mechanisms for managing change. What else have we done for managing taxonomy? From the very start of building ICMS, we have worked looking at both functionality, which we need for the collections, but also usability and supportability. And the underlying database works in a modular way so that we can maintain things and maintain the data more easily. But we present it in a common way in the front end of the system in a way that the users of the system are going to make sense. So when the living collection came along, we've had to introduce new aspects to the database to handle hybrids, cultivars, and vernacular names. We've built these in the same way as the other aspects already there, more modules that pulled in to provide taxonomic name information to the end user. When it comes to creating and adding new hybrids or cultivars to the system, we've tried to avoid some of the problems we've had with legacy data in the sense of typos, misspellings, by providing forms that enable end users to produce, to add these new categories, but using expression builders so that they're always as standardized as possible. So we're allowing for these more complex taxonomic names to be included, but in a controlled and managed way. Using these measures together, we're able to accommodate some more complex taxonomic names. I'm not going to try and go through all of this, but these have all been created using the builder so that what we have in the system is as consistent and managed as we possibly can make it. So just summarizing, it's worth making the effort to try and keep to a standard taxonomy. The future of supportability and usability of your system is going to you're going to benefit from having some of that in place, and hopefully we can avoid some of the problems we've had with previous legacy systems. We have mechanisms for keeping current so that we can look at objects across our collections with consistent taxonomy, but we also have kept fields for maintaining the historic data about previous naming. And finally, aiming to build the system for the users, whether that's the end users who want to see the complexity of the taxonomy, or us as the supporters of that system. In both cases, the easier it is for for the system to be used, and the easier it is for us to support it, the better chance of having a high quality data in the system. So thanks very much for listening, and a few thank yous for other people who've been involved in the work.
Hi, thank you for your talk. I'm doing something very similar at my collection, which is associated with a botanic garden. And so we do have a lot of cultivars that are called like a water lily that's Denver's Delight, and there's no real like genus species with it. How could you kind of like go into a little bit more detail about how you are kind of categorizing those things in your database?
I'll give it a go. Yeah, like I said, I'm not a taxonomist. We start from the we start from the scientific name. So we may only have a genus, we may have a full scientific name, but we give that as the base for the. So any cultivar we have will be linked to a full an accepted name, if that makes sense. So in the expression builder I was talking about, the first thing you do is provide a scientific name. And then from that, you can build the cultivar name.
Hi, thanks for your talk. I was interested in the user experience. Do you have surveys or some kind of metrics to track how people are actually experiencing the use of these systems? Because it's not, as a developer, ever too easy to know what the user might experience when they're using these kind of systems.
No, I agree with all of that.
Maybe you should.
Yeah, we're not a huge community in the sense of users. We do have several hundred users, but they all know who we are, and we don't need a feedback survey to get feedback from them. Yeah, so we're always looking. We have regular meetings where we provide end user support, and in those we have a lot of flexibility within Earthcape to tune the system. So we will pick things that come up. We will try and address and change those aspects.
All right.
We still have time for a couple more questions. Well, sure.
Yes, sir. Thank you.
It's great. Okay.
Hello and good.
Next up, we have a virtual presentation from Dr. Nora Lintiga Moss.
Good morning. I'm Nora Lenke-Maas and I'm a digitization manager at the Natural History Museum of Berlin. I will talk about the digitization of natural history collections in the last couple of years at our institution. So this is what the collection discovery and development has been planned within the future plan framework from 2020 to 2028. It starts with ensuring the proper conservation, because 80% of our collection have been hosted in the historical cabinets. We are going to digitize and catalog those objects and then make them accessible at our webpage. We have over 30 million collections objects, so we have to be creative how we can facilitate that within the future planned time. We started a couple of projects, but then it became clear that we needed something like a practical guideline for digitization in the natural history collection. So this is what it looks like. And I find especially helpful the cornerstones of digital collection development, which you can see here. Are basic questions that we ask ourselves together with the curators of different collections. And if you plan digitization within your collection, but a lot of those questions are unanswered, then it sometimes makes sense to do a pilot project in those collections. So this is what the collection discovery, so our approach looks like in the museum. Probably not too different from other ones, but we start with the physical condition of the collection objects. We do restoration and conservation efforts. We inventory the objects, not only with historical IDs, but we continue to use them in order to be consistent there. But we also introduce URIDs, uniform and independent identifiers with a QR code that are machine readable. Then we digitize, so taking pictures of some of those collection objects. There are quality assurance steps for the digital assets, for the physical standards, but also for the metadata, and then we want to have them in new storage facilities. And all of this process can be user-driven. It is specific to certain object groups, and there can be a type of technological development within this. If we now take the first part of this process, I want to show you one of our smaller project that we did in the Mesophyticum, which is part of the Paleobotany collection. There was a collection move coming, and we thought what would be a nice idea to capture metadata or capture information about the collection objects that doesn't take too much time, because the move was already going on. And we took the ODK app. It's a smartphone app where you can set up tables that can be filled. And the tables were set up in the following. So first you scan the cabinet QR code, the drawer QR code, and the specimen QR code. And then we have been able to take pictures from front side and labels, and if necessary, also from the back side. And then there have been some parts of object information. So is there an historic inventory number? Yes or no? Is it an original? Yes or no? How many objects belong to that number? And how is the conservation status? And that really made sense, and it had a good effort, not too much time to spare, but the move was coming faster than we expected. So we haven't been able to do that for the entire collection. Another example where we concentrate more on inventory and on the digitization part is also from the Paleobotany collection. And this is the silicified collection. There we built it, more or less normal repro station, but from the collection heads, we knew that the special part about this object is that they are polished from one side. And so usually they took, so before that, they took flatbed scanners to digitize those collections. And we got inspired by that, because from the flatbed scanners, the quality was not sufficient. So we built the repro station upside down. We cut it a hole into the table, placed a window there, glass front there. And so we have been able to produce really good photographs with a nice quality in an upside down manner. So this is a result that we have here. If we now go one step further and take this whole process into account, so from the physical object all through to digitization, but with user-driven demands and technology development, we come to my favorite project, which is the DORA project. So here we are in the malacological collections, where we have about 250,000 lots that consists of usually many objects. We think we have about 7 million objects in this collection, so we had to be creative to digitize them. So we teamed up with the Fraunhofer Institute, and they set up this nice station we termed DORA. This was the name of a female scientist in our collection, Dora Godan. This is what Dora looks like from inside. We have a label digitization part, which is a one-shot camera. And on the left-hand side, there are specimen trays with 3 cameras that are able to photo stack those images. This is what Dora Station looks like when operated. You have a drawer that you can pull out and load and unload. And this is the software that you use here. On the right-hand side, you have you see the previews of the object fields and the label fields. And on the left-hand side here, you can enter metadata. Those metadata will be stored alongside the images with the same name in the file name, and those are JSON files. Here you see the results on the left-hand side from 2 specimen objects, which are photo stacked images here, and on the right-hand side from the labels, and this is an overview about the JSON file that is stored with the pictures. And here you see the digitization with Dora in numbers. We have been able to process metadata from 128,000 lots, and additional 25,000 lots have been processed with the Dora station. So we have pictures of them already. Leaving those numbers behind, we have to capture metadata from another 31,000 lots, and 25,000 are to go with the Dora station. So in total, those are 210,000 lots, as I said in the beginning, and we processed already 154,000 lots of that, which makes a total of 73.2% of the collection that has been digitized now with the end of April. So coming back to, oh no, Dora, what's possible? Digitization, we tried out several objects here as well. So we started trying out with alcohol material, which works pretty well. We are just lacking some of the insert trays. They should be produced soon. We also did paleontological objects here, like a sea lily and a small dinosaur skull. We have objects from the archive, which are small radiolarian displays. And here we had people from the Egyptian Museum visit us. And so this is what Dora can process as well. It's not just for mollusks. If we're now coming back from the Dora station and see, like from our title, digitization of natural history collections, the more the merrier. So in terms of collection of metadata, it is absolute the more the merrier. So collect as many collection objects as possible regarding their metadata, because the more of those objects are findable, the more details can be attached in a process. But the first step and the hardest labor is collecting those metadata in the first place. And here, in terms of the amount of metadata that you capture, it's not the more the merrier, because we should concentrate on the minimal data set that is used or that facilitates the findability of those objects, and also plan the capture of pictures wisely, because it takes person hours and it also takes digital space. So what is the take-home message here. Usually you should plan your digitization efforts for the 95% that are easy to capture rather than the problematic 5%. You should check your resources to facilitate what needs to be done before you start in terms of staff, technology, consumables. And if it is possible to define different or to combine different efforts like collection moves with inventory and digitization, please do so. Also be creative, because sometimes the traditional equipment can be sufficient if you think a little bit out of the box. But sometimes also the technological development helps to spare time. Otherwise, simple, keep it easy. That's the best. Prefer online-based tools, because in our museum we had a major cyber attack and we lost a lot of the stuff that has just been saved locally. And the introduction of machine-readable identifiers usually helps to avoid mistakes. So thank you much. I hope this talk gave a little insight into what we did within the last couple of years. And if you want to reach me, you can drop me an email via this address. And otherwise, thank you for hearing my talk, and have a nice conference. Bye.
A virtual presentation. We will not be doing a Q&A, but feel free to email the author if you have any questions. Up next, we have another virtual presentation, and then the remainder of the presentations will be in person. So this next one is from Laurens Hogeveg.
I'm leading the AI team at Naturalis, and in this talk I will discuss the role of modern AI in collection management. So we all know that natural history collections offer valuable insights in biodiversity for biodiversity research and conservation. The biggest challenge in managing these collections is its taxonomic identification due to the vast number of objects, the scarcity of expertise, and missing, inaccurate, or outdated information. For example, in this paper from 2011, a 2% error mislabel rate was reported in a collection, and probably this is even larger in many other collections. We think that AI models can assist curators and collection managers with these tasks. So what do our end users need? For one, they need good general purpose AI models, right? So many projects require object segmentation or family level identification, and they should work on different kinds of images. We also think that it's very important that models have detailed descriptions, including a report on their performance, the training data that was used and how it was annotated, the intended use of the model, and of course also whether you can use the model for free in an open way. Furthermore, it should be very easy to use, and the model should scale well to large datasets. In this talk, we will focus on some new things like specialized models with short cycle updates and intuitive tooling to work at scale. So let's start with foundation models. What are they? They are general purpose models trained on large datasets. They should have good generalization, which means they can be used for specialized tasks easily with limited or no retraining. This is crucial. And nowadays, often these models are multitask, so they can do, for example, classification, detection, segmentation, and reasoning. And they are multimodal, so they work on different kinds of modalities like image, text, and audio. As an example here, you see this is from a public open model. You see how it works for automatically reading a receipt and doing some reasoning on the receipt. But we're talking about biodiversity, and also this kind of work is being done here. Here you see the identification of a yellow garden spider. On the right, you see how a human could interact with the computer to get the identification very precise. At Naturalis, we also have experience with foundation models. So, and one of them is what we call the multi-source model, or MSM. This is an image identification web service for field observation in Europe, which is now roughly used 80 million times per year by hundreds of thousands of citizen scientists. The foundation model is trained from 42 million labeled images of 5 European partners concerning about 45,000 taxa from the macroscopic life forms. So we do a few things slightly different than others. For one, we have partner specialized models. This means that the models are specialized for the partners, but these are derived from the foundation model. Also, the partners get their results in their own taxonomy. Why do we do this? Well, for one, not all partners follow all taxonomic revisions, right? So, for example, plants and fungi are often revised, and they might not want to go there directly. But more importantly, from our perspective, is that when you work with multimedia and you don't have the physical specimen, you might need multimedia-specific taxa. For example, on the right, you see Acondicta tridens and Acondicta psi, which you cannot separate on images alone. So to label them, you would need sort of a paired taxa, like Acondicta psidens do. To do all this stuff and to make this mapping and work with the data, we have developed an open source tool for taxonomy validation and mapping. And the goal is to map taxa from different sources to a shared taxonomy with the purpose of training machine learning models. As I said, it's an open source Python library, and we used it to map the taxa from 5 partners to each other, working with hundreds of thousands of taxa, with the goal of being able to pool images across partners for training of the foundation model. Just to be sure, the taxon map results are approved by the partners before we start training the machine learning models. The final result is that the outputs of our service are provided using the partner's own taxonomy, something which they told us they very much like. So now we have this foundation model, we move to the specialized models. Specialized models are models tuned to a specific context. So like a taxonomic group, a type of acquisition, a time period, a collection name, etc. On the right, you see an example of the Van Groenendaal-Krijg collection, which has about 500,000 day butterflies collected in the 1930s on Java by Jan van Groenendaal and Adriaan Krijger. And importantly, they are photographed with closed wings, which is quite uncommon in the rest of the world. Typically, these specialized models have limited size datasets from about 50,000 to 500,000 images. So if we have such a specialized model, how do we train it? Well, we propose to do this in short cycles, or what we call a short cycle specialized model. This means that we annotate, train, evaluate, and publish this model in short cycles of hours to days until user expectation is reached. If the user expectation isn't reached, we can abort the project in an early stage and don't waste too much work. The benefits of this is that the model will be optimal for this particular use case, human annotation effort is reduced, and very importantly, it reduces the user experience expectations gap, meaning there will be less, meaning that there will be less difference between what the end user expects and what the model does. So all this, of course, needs to be made available, and this is, for one, why there is DISCO. DISCO is the distributed system of scientific collections in Europe, which will ultimately contain about 1.5 billion specimens from 200 institutions and 23 countries in Europe. And this, besides all kinds of other services, it will also allow users to run machine learning models in the form of what they call machine annotation services, or MAS. This makes it very easy for end users to pick a model, run it against the dataset, and see the results. So how do we think that this loop with the human and the machine will look like? So this is the ID. On the left, you see an expert, like a curator or a taxonomist. He annotates the data. This will be stored in a database, the human annotated database. And as said before, we will then train, evaluate, and publish an AI model. This AI model can be used for multiple purposes. For one, it can be used to suggest media to be annotated, called active learning, and for data exploration. Also, it will give a set of images which have likely correct AI results, meaning that we can put them in what we call an AI annotated database. So this gives preliminary results done by the AI, and this also allows the expert to explore this annotated data already. Putting these two together, the human annotated database and the AI annotated database means we have a big database of annotated images, which can be, for example, used for e-learning modules to teach new taxonomists or citizen scientists to do annotation of data. And we think it's very important that we also use AI here. The AI can assist in picking the images which need to be revised after annotation, etc. So the final part of this talk will be about human-machine collaboration at scale. And one part of this will be what we call data exploration, especially the power of interactive visualization. So what you see here is a data set from the Van Groenendael collection of about 8,500 images. We have the true labels here, and on the left you see a picture where every dot is an image and the colors are the true labels. So now let's zoom in a little bit on one of the clusters you see in this map. This cluster is of Papilio, Helenus, and Ganius. And if we zoom into it, we see that there are some of the dots are actually differently colored. So if we look on the right, we see the actual images for this set. You see they all look the same, but some of them have different true labels, like this one. And so let's check this Graphian Sarcodonicianus. But this is a completely different butterfly if you look at it at the other examples. This means there are errors in the true labels. So what we now have done is use data exploration and visualization to detect problems with the labeled data set. These kind of techniques can be used for large data sets, and we like to scale this up. So what we envision is that we start at what we call the kilo scale, right? So this is from 32 to 32,000 images approximately. And this is what most starting annotation projects will look like. And for example, the Van Groenendael pilot was 18,500 as I said. And many PhD projects that involve data collection will have similar sized data sets. And we think those kind of data sets are ideal for rapid, short cycle specialized model development, like hours to days. But then we move to the mega scale. Mega means, in our terminology, from 32,000 to 32 million images. And now we enter the large museum collections, like the Van Groenendael collection, 500,000, or the Smithsonian Herbarium, 3.8 million images. Here, exploration and efficient labeling strategies are really required. But we want to move on and to the gigascale, or billion scale. This means about 32 million to 32 billion images. And for example, all museum collections in Europe together, which are in DISCO, are about 1.5 billion specimens. And this is also, by the way, is another example of the scale expected coming from continuous digital monitoring programs every year. So this will increase a lot. And here, really new novel approaches are required, and this is what our team will focus on in the coming years. Thank you, and the room is open for questions.
Okay, really interesting stuff. Thank you to Lawrence. Up next, we've got an in-person. What's up? Oh, okay, thank you. Up next, we've got Marisa Sorovi.
Hi, good morning, everyone. I'm Marisa Servi. Is this right? Okay. I'm Marisa Servi. I'm the museum specialist for mammals at AMNH in New York, American Museum of Natural History. And I'm here today to talk about a little bit of a different kind of project that has been not talked about this morning, but this is a smaller scale data management process that we're doing in our department. So it's about our annual fieldwork we're doing in Belize, catching bats, and the data collection and how we've streamlined that over a continuous reoccurring field season. So each year, our curator, Nancy Simmons, along with 2 mammalogy staff members, lead a 2-week field season to Belize to focus on capturing and collecting data on bats. The work is done at Lamanai Archaeological Reserve, a significant Mesoamerican site that was once a major Mayan city. What started as a small-scale project has evolved into a large-scale collaborative effort involving over 80 researchers over those 2 weeks from various institutions and countries. Everyone comes with different questions related to bats in the neotropics. Over the 15 years, the work has trained many students, has contributed to numerous publications and has numerous publications and resulted in collections for nearly 30,000 samples over many different projects. Just since 2021, our 2-week field season has averaged a capture of 900 bats and has collected over 2,000 samples in those 2-week periods. AMNH alone has added 700 specimens to our collection, 7,000 tissue samples, both lethal and non-lethal, to the mammalogy collection over the last 15 years. As you can imagine, all these people, all that data, and all that bats are a lot to manage. So that's our biggest challenge. Many researchers are focused on their own research, and so when it comes to collecting the data accurately, it's often not done well. So we've made a streamlined way to manage that data with using different forms, so it's foolproof and standardized for everyone in the field. Our goal is to make sure that people know exactly what to fill in, fill out, and so there's no questions. Researchers every night head to the nets at the Lamanai Reserve. They set up nets for catching bats, both harp traps and mist nets. They are then given a dry bag that's filled with white cotton bags that have individual numbers and letters distinguishing between them. These bags are specifically, designed to identify fecal samples that might have been placed in the bag, or any ectoparasites that might have hopped off the bat during collection. Also in the dry bag with these bags is a pre-printed field data sheet that you see here. This sheet is designed to fill out easily and accurately. Researchers simply circle the net type they're at, the location they're at, and record basic data on the bats, such as time of capture, sex identification, taxon identification, GPS location of the net, any notable observations about that one bat. That information is then transcribed to a pre-printed sticker that then is applied to the corresponding bat's bag, and that is then that with the field notes, the bat with the field data sheets returned to the lab for processing. So proper identification of the bats was another huge challenge, especially for researchers who have not worked in the neotropics or in Belize or the site that we were working at. So Nancy Simmons and her colleague Beth Clare developed a laminated field guide for identifying the Belizean bats that were catching at Lamanai. Copies of the guide are available at every single net. They also have, they're bound, and they have a plastic ruler for easy measurements of forearm and any kind of foot measurements that would distinguish the bats. Each page has highlighted distinguishing traits to help researchers accurately or close to accurately identify the bats. So while the guide standardized the naming identification in real time, we still had a huge room for improvement on how we were maintaining numbers. So before 2019, researchers would be assigning their own field number when they were sampling from the bats. This ended up being that there was no central system or coordination. As a result, a bat could get multiple IDs or multiple numbers. There was no way to track the collection of the samples or who took the samples. There was no comprehensive log of the captures, making it nearly impossible to track recaptures, collect data across researchers, or understand the full scope of what we were collecting over multiple field seasons. That changed in 2021 when a researcher team from the University of Waterloo proposed a new approach focusing on monitoring bat recaptures. The idea was to simply implant a microchip in the bat, the same way we do our dog or our cats. This PIT tag allows researchers to scan and instantly identify each bat, making it possible to track recaptures in real time and to gain valuable insights into population dynamics. With this major shift in methodology, we also had the opportunity to revamp our own numbering system and develop a standardized pre-printed form to help efficiently process the large number of bats coming into the lab. In 2021, we launched the BZ numbering system. Each bat captured is assigned a unique BZ number that stays with it for its life regarding who handles it, who takes samples from it. When the system is simple and sequential, beginning in 2021 with BZ1, and we've already surpassed BZ 4000. This consistent numbering system allows easy tracking of recaptures whether it occur in the same field season or years later. For example, the vampire bat you see here was captured the first week of 2024 field season and assigned BZ number 3265. And when it was caught again the second week of that same field season, it was assigned its BZ recapture number of BZ 3265.01, linking instantly to the BZ number. In collaboration with the University of Waterloo, we decided that as part of the pit tag project, it would be beneficial if we were collecting non-lethal samples from every single bat. So hair and wing samples from every bat that was being processed by the pit tag crew. These samples were then brought back to the AMNH and accessioned and added to the mammalogy collection, allowing researchers to study the same bat over multiple different years of its life. To support this effort, we worked with the Waterloo team to design a data sheet that captured both the required information they needed for their research, along with the information we needed for our cataloging efforts. These forms are designed easily to use and accurately, like the field data sheets that we have in the nets. Researchers simply circle or fill in predefined fields. The forms include sections for BZ numbers, taxon ID, sex ID, reproductive status, measurements, and it has even a designated area for the pit tag barcode sticker that comes with every single pit tag equipment and has that same number that is associated with that pit tag. These are specific. There's even specific spaces on the form for hair and wing that have already pre-printed markers for hair and wing. So researchers only have to fill in the vial number that the samples are in. My two favorite parts of this form is a section listing the box number, which indicates the box that the samples are in. This allows me to make sure that everything is in the right box. And my last favorite part is at the bottom where people have to sign their names so I can track them down because they have poor handwriting. So a lot of these people are sleep-deprived researchers who have been working very tirelessly every night. And so they tend to, once again, not clearly state what they're writing, and I will track them down because they can't write a 9 or a 7. So everything that's collected goes onto our master spreadsheet. This is compiled daily by the AMNH staff. It's controlled only by us. No one else is to touch it. It is our central record for every single bat that was processed since 2021 and allows us to confirm recaptures quickly and, by the pit tag number, identify and flag any kind of inconsistencies that we've found across multiple years, track every sample by box, slot, and vial number, and it helps us maintain compliance with permits and customs regulations. Since 2019, the mammalogy team, the mammalogy department has taken on the responsibility of exporting all the samples taken in the field season under one permit. So that includes not only the stuff that's coming back to AMNH, but every single researcher's samples that is being collected in those 2-week seasons. Centralizing process ensures a smooth compliance with customs regulations for export and import into the U.S. But this change has introduced two major key aspects to our work in Belize, which is first, we now maintain a clear log of every single sample that every single person is taking. And then secondly, it makes us track every single location of every single sample. So that looks like we record where the samples are in the cooler, in the dry shipper, down to the box, down to the vial, down to the bin in the dry shipper. This meticulous organization allows us to provide U.S. Fish and Wildlife with precise information if needed, preventing them from delaying the shipment in process. To ensure streamlining of this process, the AMNH team has decided to store all of our hair and wing samples in desiccant. So at the beginning of the field season, we take 2,000 pre-labeled tubes from our cryo lab at AMNH, pre-labeled with barcodes and designated numbers. The box is then given a range of numbers and assigned a box number, and then that box, along with a stack of data sheets, go to a pit tag crew station. And as the pit tag crew fills up the samples, once the box is done, the samples come back to us, and I barcode scan every single tube into a spreadsheet and input all that information into the master spreadsheet and cross-reference all that information. If no errors are found, the box is then taped up and not touched again until it gets back to AMNH. Once back at the museum, we divvy out the samples that are for other researchers and participating institutions, and then we send those out and we catalog the samples that stay at the AMNH with our standardized language using our EMU database. Each recapture is given a new catalog number, so it reflects its new year, its new location, and the stage of life of that individual bat. All related records are then linked in our EMU Relationships tab, allowing researchers to track the individual bat's history over multiple field seasons. We've also reevaluated our storage strategies in our fluids preserved collection, especially the taxon that we collect multiple years in Belize. While standardized practice has been to jar everything by species, location, and collection year, with repetitive use for the last 15 years of collecting, we've come across space limitations. So to address this, we've begun consolidating specimens from multiple field seasons into the same jars, freeing up ample space for future collecting. Finally, all the data, including the data sheets, the BZ sheets, the field sheets, and the master spreadsheet are all scanned and archived, and then they're made available to any researcher in the Belize project for cross-reference and check. Okay, thank you. That's if you guys have any questions, happy to have them. Cool. One question.
Wait until,
Oh.
sorry, that was loud. Please wait until a microphone comes to you so that we can all hear the question and the people online can.
Who is asking? I
Yeah.
noticed that the field tags don't have a section for researcher. Was
Yeah.
there a reason behind that?
Yeah, so we've just kind of centralized it to be that everything is collected under Nancy Simmons, which is mostly, I think it's a reason for permitting. The actual individual researchers who are taking samples for their lab and it stays in their lab, they would actually just have the BZ number as the field number. And so in our records, it's Nancy Simmons who's collected, but in theirs, they could have the Dan Becker lab or any other lab that they're working under, but the BZ number would be their field number and their collector number. Yeah.
Order out of chaos. You're my kind of people.
Yeah.
Thank you.
Yeah.
The pit tags,
Yeah.
what do they cost individually?
Ooh.
And are there any like additional sterilization procedures you have to do with the tagger?
Okay, I don't know cost because that is above my pay grade, but I can get that information to you and I'll get you my card. The sterilization, they're already pre-sterilized. They come in like medical grade sheet, packets that you rip open and, use and then dispose of. And they also come, I know they come in different grade sizes, so you can use them. We have like the smallest little ones for bats. Yeah, hope that's helpful. Cool.
There is a question from online.
Impressive. Okay.
Where is the master spreadsheet stored? Do you have any backup?
Oh, okay. Is that someone from the Belize Project? Okay. Yeah, so it's all stored actually on the, it's first stored on our hard drive at AMNH that we have, but also it's on the, it's on the Dropbox. That is a shared Dropbox that we have for the entire project. And it is locked and not editable. So if anyone wants any changes made or they have any questions, they must contact one of us. It is, it's a very controlling chaos situation. So we don't let a lot of people touch those things, but yes, it's available for people to search and look up. Okay, cool. Thank you, everyone.
Thank you to Marisa for a great presentation. Up next, we have Allison Wilkins coming all the way from Utah on barcoding museum collections. Okay, take it away, Allison.
Cool. Well, nice to see you all here today. I have to say, all this talk of theses this morning is giving me strong flashbacks to defending my thesis in this building. So if you have revisions to anything you see in my slides, please don't send me an email after the presentation. All right, so I'm going to talk to you yet again about barcoding, and because this is the way my brain works, I'm going to first tell you a million fun facts you need to know about barcoding, or that you'll never need to know about barcoding in your life, but could win you Jeopardy, in which case, please split the prize with me. So what and why are barcodes? Why do we use barcodes? So barcodes function. It's more complex than this. Please don't tell my tech friends that I'm telling you this. Barcodes are basically fonts in Word documents and mail merges and things like that. They were invented in 1948 and inspired and derived from Morse code. That's why there are the spaces and those line differences. And that's why if you do a linear barcode, when it gets really long, it has to be a very certain size. But they weren't really commonly used outside of the auto industry until the 1980s. Museums are early adopters, as we always are with pretty much any technology, both to our benefit and to the bane of our existence. I don't know if you've ever used some of the really weird film copies, microfiche. If you have questions about that, ask me later, or don't, please don't. The latest barcode innovations are basically 2D barcodes, so combining barcodes in different directions. So rather than this linear format, like this lovely zebra, which is apparently what tech people call barcodes or zebras. And I'm like, oh, zebras? We work in the dead zoo, zebras in the dead zoo. Correlations, everything goes back to natural history. And everybody's innovating barcodes. QR barcodes are some of the more recent innovations. The guy who invented them is still alive. He's very nice. So yeah, barcode font choices. So you can find these. These are some of the technical names for barcodes and what they look like, in case you have forgotten. In which case, I've got many questions for you about how you forgot what a barcode looks like. Moving on. So I'm here to tell you today how we've used them for the most part. And so internally within our museum system, we use primarily EMU, Arctos, and Symbiota. And so this use case for both anthropology and paleontology is within EMU. And so one big problem we all have, I'm pretty sure, is what do we do about all the unprepared materials? And when you get an email from a scientist who worked with you or your boss 15 years ago, and it's like, I want to know how much material we have left that we did not look about or care about those 15 years ago from this one specific site. And you're like, well, you never came back to research this. What are you doing? Asking for all this material and all the things that keep getting added to this. So this is one of our unprepared aisles, one of them. And so everything comes in plaster jackets, pallets, boxes, bags, newspapers, beer cartons. Yeah, you know who you are. Generally all with vaguely known sites and collectors. And, we're always left with the question, how do we know what's left? How do we know that, Adam Smith from California, who wants this one tiny fish fossil in this 20-pound bag, actually find it? Can we find it? In which case my colleague can tell you that yes, we did find it. So the first step is how does our computer system understand locations? So please do not judge me that the barcode field I could not get to work in EMU. And so our barcode is basically randomized gibberish of a generic geo number. So UMNH geo 30 in this case. I did not center anything in the images, for which I apologize. Sorry, center anything on the physical cabinets. And so you got your standard location hierarchy, which is the museum, the room, that's the unprepared row, and then the various columns and the shelves tied to that columns. And so after much trial and error, which is longer than this talk can care to last, we decided to treat anything that is a fixed location as basically a location. Words are meaningless when it comes to computers. And then everything else that is a box or a movable unit is a holder. And so that way, when our prep lab manager comes up, reappears out of the ether, takes something and moves it, he said, and I don't have proof of this yet, that he will update this. I will let you know if that happens. And so within that, we can create individual catalog records that share that temporary location barcode nomenclature. So that way it can go away when eventually this is a fossil, which I am not entirely certain this isn't just rocks. But I'm not a geologist or a paleontologist, so not my monkey, not my circus. And so that way, that this object, which shares that barcode number, because I really didn't want to reprint 3,000 more barcodes, can have that field information and that location and be updated in real time. And so this was like our first test case to make sure that we weren't completely crazy and that, yes, we could convince our less than technology-friendly colleagues to actually use barcode scanners and like Excel spreadsheets. And then the apocalypse happened, which if you want to hear more about this, please go see my colleagues talk later, of a sudden donation of like, what, 2,000 Cambrian fossils in a house that was sketchy. Yeah, definitely not a common occurrence, right? And so I think I got 2 days notice that suddenly we had 4 days notice to move everything out of the house. And I'm like, cool. How? How are we gonna do all of this? So you can see that's us in this like garage in this house up in the mountain. In addition to all the fossils, we had like 20 years worth of their entire body of research, both related to the fossils and unrelated. Researchers, please take this as a plea. Go through your data. You do not need to keep everything. I've seen things I cannot unsee now. Oh, I've got some stories. Ask me about it later. But including every single file format you can possibly imagine. So everybody's heard of, floppy disks, hopefully. I've had some students that just, they think it's the save icon, which it is, but Jazz drive, hard drives, photos, slides, documents. And so how do we keep track of how all these things relate to each other and how everything in the house relates to the house itself? And so, yeah, how do we keep track of, because it's important in case it gets lost in context, because they're like bags and boxes and bags and boxes and bags and boxes and so on and so on. How do we keep track of this association within itself? Because if something gets misplaced, then we need to know how it existed within the context it existed. I'm repeating myself. And so the numbers have to be temporary, they have to be movable, they have to be fixable, and apparently they have to be heat proof. Which you shouldn't put them near a heater. And so this is sort of what we had to implement in like the 2 days I had time to plan this all out. And so it's a little messy, but we use Sapphire forms, which is a thing you can use in EMU, but the real issue there is you can only work in one module at a time. So you created a location for a drawer and a cabinet, and that cabinet drawer is tied to a picture of the cabinet, to a picture of the house, to a picture of the garage that's in the house, and then tied to individual fossils in boxes as it gets moved. The boxes itself are treated as catalog records. So as they get updated, split, or recombined, then we can update the catalog number with the actual number. And I see some of my colleagues cringing in the audience. So this is why you make me do this. And so everything is movable, flexible, and adjustable. And some of it actually worked. And so, sorry, this is a little disjointed. I did not get the email that they extended the deadline to update our slides. So, our other big issue outside of this, because I'll tell you the problems and the benefits of barcoding things later, apparently, is mass imaging archaeological artifacts, which is one of our other big problems. And so we have approximately 2 million artifacts composed of about 7,500 sites collected from the 1920s to present within the state of Utah, held collaboratively, maybe, with state and federal land agencies, and with many problems to consider. How do we photograph a wide variety of objects to scale so that way one student isn't stuck in the photo lab photographing fire-cracked rocks for the 50th hour that week? Unless you want to drive them insane, which is kind of funny sometimes. And so I'm like, why don't we just use barcodes, make my life easier, because people tend to make errors. We're not machines, as despite what people may wish. Our original system was a combination of mail merging and copying and pasting. So if you can imagine copying and pasting fields for archaeological artifacts when you're like 300 in, you will hurt yourself, FYI. And so the bottom is our old labels compared to our new labels up top, which automatically pull from Emu. But as I was going through and making a student rename these, because I was not going to rename 1,000 photos. Not that I didn't do that, because I realized afterwards that having a student type in file names can be messy. And scientists are also very messy when you run into duplicate catalog numbers. And so that was the biggest thing we learned, is that not all catalog numbers are unique, despite what people want them to be. And then that causes a lot of problems if you're trying to move things automatically into your database system. So that was fun. And different number of formatting styles historically. So sometimes there's a space, sometimes there's not a space. Sometimes there's an underscore. Please be consistent. I should have added more slides. And so we're going back to this barcode on the top, which is a data matrix style barcode. It was small, it's easy to slap on things. It does require a separate computer system to encode the information. So now we're doing it one by one, and we've done approximately 40,000 automated file renaming systems and import systems to get all these images into our database. They're mostly lithic, so they're the same size and shape. It's easy to do. You don't have to change the camera setting. And so the next step is we're going to split on a grid. And I was going to put up an example of an archaeological example of this. And then I realized I put it into our system already. So I do not have any holistic images. So enjoy these rodents. And so basically what we have now is a system in place that was developed by our IT staff to take an image like this, and you program in columns and rows, and you can split it and rename it based on that. And so it's one process from beginning and end to split, rename, and or combine if that's something you want to do. Because everybody wants something different. So some of our mass renaming pitfalls in excess. Don't use catalog numbers if you don't know they're unique. And just assume they're not unique. People make mistakes, and that's okay. Duplicate numbers, inaccurate historical information. If you do linear barcodes, they get huge real fast. So unless you want 4 or 5 inches, do not use linear barcodes. The lack of initial buy-in from all users was a big problem. You know who you are. Barcodes aren't aesthetic was actually a comment I got. They don't look pretty. And emu barcodes is interesting. Barcodes work really well in Arctos and really well in some parts of Symbiota, but integration with our emu collections management database is complicated. And so this is sort of the really janky workflow of how do we get it from taking a picture, renaming the file, ingesting it in emu, ingesting it into our digital asset management system, and putting it back. I think that is a subject for another talk. So please ask me questions. But yeah, otherwise it's going pretty well. We've had a really small margin of error with our move-based barcoding system, with only about 20 to be fixed. That's not including, for some reason, our database isn't putting unique identifiers on any of our catalog records. I have a ticket out for that. More experimenting is needed from data matrix barcodes and data matrix papers. I know there are a variety of different papers that can be used to compensate for heat and alcohol. And then, yeah, barcoding just allows for things to be made easier. It can also allow you to compensate for things that you just don't know or want to know, you want them to be known to exist. And so if you have questions about anything other than the computer stuff, please email my colleagues. I am like 90% certain that this is a QR code that leads to something and not just a picture of a QR code. I think it's to search our digital collections where you can see how these barcode images are turning out and how they're being made searchable outside of our internal CMS. So thank you.
Hi, also get you chaos and annoyance. My question is, what level of, or what version of emu are you guys running?
So we are running an Emu 9, but
Okay.
the previous version of Sapphire, because something is wrong with like our university firewall. So
Okay, we're way behind that, so no hope for us. Okay.
no. It's definitely possible and relatively easy. It's just really strange. Yeah. Does anybody have any question or want a fun fact unrelated to barcodes? I'm full of them, as my colleagues can tell you. Okay. Okay. Thank you, Allison. Okay, we've just got to get this next presentation set up really quick. So I'm going to tell you guys a little story, I guess. Totally not stalling for time, right? So we're all scientists. We all know the process of research comes with problems and sometimes funny and weird situations, right? So a couple of years ago, I was studying urbanization in European starlings. And part of the research was to set up this tent that we would put the birds in, right? And since I'm studying urbanization, I was at a public park. So someone went and warned my lab mates that there was a homeless woman crouching in the grass. It's me with my tent. Of course, I'm a graduate student, so I was wearing like horrible field clothes, right? Like holes in my field pants, bird poop on my hat, crouching in the bushes. Okay. So I think we're ready to go. Next up, we've got Michael Elliott from the University of Florida telling us about connecting online biodiversity databases. Enjoy.
You're good. Okay. Hi, everyone. I'm Michael Elliott from the University of Florida, and my talk is on connecting online biodiversity databases via community-driven multi-agent systems. And it's, I also should acknowledge Manuel Luciano, who works on this with me. I don't think I put him on the authors Oxford, so I feel bad about that. Yeah, so this is a continuation of work we did at last Spinach, if you were there. Way back when in Okinawa, we presented a chatbot for IDIG Bio, and this is an extension and continuation of that work. But back then, we had a chatbot that was designed to answer questions like find records of this species matching these search criteria, and then hopefully it would respond, I found what you're looking for, maybe a certain number of records. It would also point you to the actual data as it was presented by IDIG Bio, and then it would also give you metadata describing exactly what it did so that you know it's not lying to you and you can reproduce it yourself. And so now this is kind of ancient technology, hence the washed out image. But the basis of what's going on here is still the same when we carry it on. So this was called a multi-agent system, where an agent is just a program that's capable of making its own decisions based on whatever input you give it. It decides how to tackle the actual problem and what the problem is. So we had a multi-agent system, so there's multiple agents. The little tiny robots, do I have a pointer? Yeah, I do. The little tiny robot faces here, they represent specialized agents that are designed to retrieve really specific types of information from IDIG Bio. But on their own, they're not very general purpose, so you don't really want to interact with them directly. So we have this general chat agent that directs you to one of these specialized agents. And this will become very important later. But that is sort of the foundation, the conceptual design of the whole system. But we realized pretty quickly that we're not just limited to IDIG Bio. There's a whole world out there. Just biodiversity itself has probably more things than we could ever hope to implement ourselves or support ourselves. But also outside of biodiversity, things like regional boundaries and social media and all these different things that might be relevant to the data you're looking for. You might also want to be able to grab those and use them to query your data or compare against your data or filter your data. There's also non-biodiversity research that maybe you have some kind of cross-disciplinary study. We should be able to get all of that too. And then finally, maybe not most importantly, but kind of the thing that pins it all together. So what I just talked about, those three things, we're co-locating data. The chatbot gets data, it brings it all into a same space so that it's kind of ready for you to use it in some kind of comprehensive integrated manner. But then we also have these processing and analysis bubble in the bottom right, and this represents things that you can do with the data. So ways that you can filter it, process it, prepare it so that it meets your needs, or integrate it, combining it from different sources, and maybe doing some cleaning and things like that. So that's where we have the integration. So we have the co-location where we grab data, and the integration where we actually make use of it and turn it into something new. This is the vision. Back then we were just doing IDIC Bio, so we just had collections and media data, and that was about it. But hopefully someday we'll cover a much larger span of that. But where we are now is we christened it, we gave it a name, iChat Bio, and it's got a website I'll show you later. But now we support also GBIF and BHL and iNaturalist, and we provide a little bit of JSON tools for parsing their outputs. All of these services give you JSON by default when you download their data through their APIs. So we have some processing capabilities as well. So now, not only do we search IDIC Bio, but we can search beyond. You can dig into and transform the data. I'll show you a little bit, maybe later today, actually. And then the whole thing's kind of been redesigned for research, because for research, we don't, like if you go to ChatGPT and you ask it to give you something or find something, maybe it's even searching the internet, still you don't really get a good trace of how it got that information. You don't know if it's interpreting things correctly. You don't get the metadata, the attributions might be lies. So in the research application, we need to be much more careful to make the system more reliable. So the whole thing is sort of, that's why there is an iChat Bio instead of ChatGPT with, your customized assistants, is because we have these extra constraints. And most importantly for this talk, we've redesigned it to be modular and extensible. So the idea is we have these agents, but we should be able to drop in more agents later. And that's where we'll get to the community-driven iChat Bio. That is what we see as happening next. So this integrating or involving more people brings in a lot of challenges. So first of all, we need to have more consistency. We need to define requirements. So what are our reliability guarantees? If this chatbot produces some data, uses some kind of processing, how do we propagate metadata and attribution? And more specifically, what metadata, what attribution information, what fields, how do we present that in a way that is useful to people downstream? Personal responsibilities. So if multiple people are invested in the system, who's actually hosting each agent, who's running the web servers, and that all provides implies cost. There's also data providers can contribute by preparing their own access protocols to make it easier to work with things with the agents. And then the users as well, they may have some responsibility as far as correct usage and all of that. So all of this needs to be thought about. But as far as I'm concerned, I'm an AI engineering oriented person. So the agent design standards and protocols, how we actually implement this at a technical level to enable whatever requirements and responsibilities the community has decided, or I mean, we're as it evolves, we should be able to support whatever we need. So moving along, here's an example just before I get any deeper. This is just, I can't really go very deep with a single slide, but here's just a very broad use case. So suppose I ask the chatbot, I need all the record data and literature you can find for this species, which I think is a fruit fly. And it goes, and these boxes represent its calling specialized agents. So it's searching IDIC Bio, it's searching GBIF, it's searching BHL. And these little created lines means it's downloading data and assigning metadata and tracking everything it does. And then the next page over here is the continued response. It verbalizes everything it did. You can see it found a bunch of records on IDIC Bio, a bunch more in GBIF, and then some literature in BHL. And all of this links back to the processes that these agents initiated. So you can trace it and you can reproduce it and make sure it's not lying to you. So right now, it's not really community driven. It's kind of, we implemented these agents. They're for other people's services for sure. But still, it's all kind of in-house at the moment. But the benefits of switching to a more community driven approach or accepting contributions from the community is Well, first of all, looking at from the contributor's perspective, why would you want to contribute? So now we're kind of turning the presentation into a chatbot. I guess I'm the agent. I don't know. But in this community system, if you contribute, if we have all these other services and you contribute a new agent that exposes your service or contribute some knowledge or something that helps, then this chatbot, the idea is that it's a single conversational interface, and you don't necessarily need to know where your data that you're interested are, but it will connect you to them. So maybe you have a service that you connect to our system, and one of our users comes by and it says like, I want pictures of bugs or tools to process bug data, but they don't know about your service. Then iChatBio will direct them to you and also be able to interface a little bit with your service to actually give them a preview of what it looks like to use your systems. Second of all, it lowers the expertise barriers because it's a chatbot. It's natural language. You could be a fifth grader and be able to ask it questions, and it would interpret them into proper queries for actually getting the data. It'll help you reach the level of technical language that you need, at least for a specific system if you're not specific for the system, but also more broadly at a scientific level. And also bringing complementary services into a shared space. So if maybe your data complements what's in GBIF, or maybe somebody has a tool out there for processing data, if that's exposed to the system, your data can be fed straight into that tool. So there's no need for setting up your own complicated workflows. And then just in general, it gets better as a whole as more things are added. It's a scalable system. The more features there are, the more general abstract questions, higher level questions it can answer beyond just fetching data. So the more the merrier, and everybody, I think it's a real win-win if we get a significant amount of contribution. But how do you actually make your stuff? So first of all, how do you make your stuff agent friendly without even worrying about making agents? The two main ingredients are machine accessibility. So APIs are really important. These are what scripts and programs use to actually get your data. Because if your data are just available through like a landing page with like HTML with text and like here's a link, that's really hard for an automatic system to actually find that data in a reliable manner. Although it can be done, it's just not very efficient and it's prone to failure. So making your stuff machine accessibility, that's a great first step. And then second of all, thorough documentation. So these AI models that we use, they're very naive and they're very gullible. So they follow the instructions you give very closely. If you give good instructions, they will perform well. But if you're very vague, if your fields in your data are not well defined or they don't have descriptives information, even if it's obvious to a person, it really needs that explicit documentation so that it knows how to use your data to the best extent, or your services. And second of all, if you want to go a little further, how do you make your own agent? So we're working a little bit on it right now. It's all developing very quickly, but we have something called the iChatBio SDK. The vision is, you can do it right now, but I don't recommend it. If you have Python installed, you can just run this. This is the Python package importing tool, but you can do pip install iChatBio SDK, and it'll give you access to the tools to develop your own agents in a way that works with iChatBio. But don't do it just yet, and you're a real pioneer. But how it works is, in general, in the general world of AI, there are emerging protocols for letting your agents communicate to each other. So this is where I said our last year's work was kind of ancient technology, because nowadays everything's moving so fast, these protocols, they didn't exist 2 months ago. So we're having to adapt pretty rapidly. But the idea is we have our agent, the iChatBio agent, and if you make your agents that implement this A2A protocol, it's called agent to agent, then they can talk to each other. And they can also talk to agents outside of iChatBio. So it's not like you have to design specifically for us. But if you use the iChatBio SDK, we handle all of the metadata propagation and all of the finer points that let you interact with iChatBio to the fullest extent. And yeah, that is all. Thanks for listening. We have a demo later, so I didn't really get into exactly what it looks like to use it because we have a demo later. But there's my email and the QR code. If you want to talk to the chatbot, feel free to scan that. And otherwise, I hope I see you later. Okay, so we have 3 minutes for questions. And I'm pretty blind, so you can shout if I don't see you. Oh.
Do you have a plan for disseminating like information about this to like a broad public?
Yeah, so the best plan I have personally right now is we have iChat.io. So and the a lot of people know about the URL now. But we're just gonna add like maybe a blog or something onto that page itself. I know that's not the best way because you have to know about it first, but still it'll keep people in the loop, especially about development. But I probably shouldn't commit to anything because because I'm not on the media side of Idig and stuff. So I'll leave that to the people up top. But if you give me your email addresses, I will personally disseminate information to you. Anyone else? All right.
Okay, well, thank you so much, Michael.
Yeah, thank you.
And with that presentation, that concludes this session. Thank you to the speakers for some truly great presentations, and thank you to everyone for your attendance. As a reminder, there are links to on-demand viewing of this session that will be made available to all of you via YouTube. I also have a quick announcement. The silent auction and art auction winners lists are posted at the registration desk, and please make sure to claim your items as soon as you can. Thank you all again.